Varenicline nitrosamine testing. Let's start with a scenario. A widely tested drug, Varenicline, used to have millions quit smoking, suddenly comes under scrutiny. Not because it stopped working, but because of something invisible trace level impurities. This presentation is about how the pharmaceutical industry responded to that challenge through discovery, regulatory action, and advanced analytical science to ensure one thing above all, and that is patient safety. Hi, I'm Raghuveer Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories, Canada. Welcome at Resolve Mass Laboratories. We bring deep expertise in advanced analytical characterization and complex molecule development to accelerate your IND and ANDA programs. So, what exactly are nitrosamines and why did they cause such concerns? Nitrosamines are chemical impurities that can form when amines react with nitrosating agents, especially under certain conditions like acidity or heat. What makes them critical is this: many nitrosamines are classified as probable human carcinogens. And here is the key point: even in extremely small amounts, these can pose long-term health risk. That's why the detection and control become a top priority across the pharmaceutical industry. Now, why was Varenicline specifically at the risk? The answer lies in its chemistry. Varenicline contains an amine group which makes it susceptible to a reaction called nitrosation reaction. This can lead to a formation of specific impurity known as N-nitroso-varenicline. But this doesn't happen randomly. It can be triggered by residual solvents, impurities in raw materials, degradation over time, or even interactions with excipients in the final formulation. So the risk isn't just in manufacturing, it can continue throughout the product life cycle. Regulatory turning point. Once this risk was identified, regulatory agencies like FDA and European medicines agencies took immediate action. They introduced strict intake limits, often in the nanogram per day range, but more importantly, they made it mandatory for manufacturers to perform detailed risk assessments. Every step of the process, from raw materials to final product, had to be evaluated, and validated analytical methods became a global requirement, not just a recommendation. Now comes the technical backbone. How do we detect these impurities? The most widely used technique is LC-MS, MS, or liquid chromatography coupled with tandem mass spectrometry. Why this method? Because it offers extremely high sensitivity, allowing detection in the nanogram per ml range. It's also highly selective, meaning it can accurately identify nitrosamines even in complex drug mixtures. This level of precision is essential not just for compliance but for confidence in the results. API versus finished product. Testing the active ingredient or API is relatively straightforward. There are fewer interesting substances and sample preparation is really simpler. But when we move to finished product, things get more complicated. Excipients can interfere with detection, nitrosamines can form during storage, and each formulation requires its own validated method. So, while API testing gives us control at the source, finished product testing ensures safety all the way through to the patient. Now, there are some lab challenges. So, in real lab conditions, several challenges come into the play. Achieving very low detection limits is difficult but essential. There is also a risk of performing nitrosamines during the analysis itself, what we call artifacts. Matrix validation becomes complex, especially with multiple excipients, and even reagent purity matters. Trace contamination can affect results. That's why analysts must carefully control conditions like pH, use nitrite-free reagents, and follow strict protocols to ensure accuracy. Now let's talk about risk mitigation strategies. But testing alone isn't enough. We need prevention that starts with reviewing the synthetic process to eliminate the nitration risk. Raw materials must be strictly controlled to avoid contamination, and even packaging and storage conditions needs optimization to prevent degradation over time. In simple terms, the goal is to design a process where nitrosamines never get a chance to form. Now let's talk about industry implications. What does all of this mean for professionals in the industry? Whether you are in a QA, QC, R&D, or regulatory affairs, nitrosamine control is now a core responsibility. There is no longer a niche topic. It directly impacts product approval, regulatory compliance, and most importantly, patient trust. Mastering this area is not optional anymore. It's become the real gold standard. This brings us to the end of the presentations. So what's the takeaway from this Varenicline case? It reminds us that even well-established drugs can face unexpected challenges. But with strong science, robust analytical methods, and proactive risk management, those challenges can be controlled. At its core, this is what pharmaceutical science stands for: precision, vigilance, and unwavering commitment to the patient safety. If your program demands precision, expertise, and regulatory-ready data, contact Resolve Mass Laboratories to support your complex drug development journey. Thank you so much for watching this session. Goodbye for now, and see you in the next one.